Hey guys, welcome back to my channel. So today I want to talk about GTPase. So this concept is really related to the last video I made, G protein coupled receptors. So you can really find it in my biology playlist. So this video really fits into that video, okay? So I hope you can watch both. All right, so back in the days when I was in my cell biology class, a lot of students were confused about what's the difference between GTPase versus G protein, right? What about GEF and GAP? What do they do? Yeah, it seems like most people, when they first learn this, it's not very intuitive. So the only thing they know that they're surely aware of are GTP, GDP, right? They're like, oh, I know what is GTP, what is GDP, but really a lot of the terminologies are just confusing people. So we don't want to linger on any confusion when we're at the point studying MCAT. So let's just clear up everything, okay? All right, so GTPase is really the biggest word, okay? So under GTPase, I break it down into two categories, okay? One is called small GTPase, okay? And then these are the proteins that they have a single subunit, okay? They're small GTPase, they're small proteins. And then the other category is called G protein. So in my video, G protein coupled receptors, I talked about the G protein that has three subunits, alpha, beta, and gamma, right? So the differences between the small GTPase versus the G protein are the structural differences, right? One is being simple, one is being like more subunits. That was the only, difference that I can think of at this point of my education, all right? So for small GTPase, there's another name called small G protein, okay? All right, so I hope it's clear, okay? I know it's confusing how biologists name things, but yeah, we get it, okay? All right, so we also learned that we can either turn on or turn off GTPase, right? So when GTPase is bind to the GDP, then we know it's the off status, right? It's inactive GTPase, okay? When it binds to GTP, we know it's on. We turns it on, right? It's an active form, okay? But how do we go from the inactive GTPase when it's binding to GDP to the active form when GTPase is binding to GTP? Okay, so there are two other proteins that comes into play. One is called GEF, GEF, right? Another is called GAP, GAP. Okay, so go from inactive to active. What do we use? We use GEF. Okay, so what does GEF stands for? GEF stands for guanine nucleotide exchange factor. Okay, so what does it mean? Like, it literally means it changes this GDP into GTP. Okay, it's not adding a phosphate group. It's not adding a phosphate group to to turn the GDP into GTP, but it's literally like it gets rid of the GDP and then exchanges it with the GTP. Okay, all right. So once the exchange is completed, then our GTPase is in the active form, right? Okay. Now, how do we go from the active form back to the inactive form? Okay, so this is GAP. Okay, so what is GAP? GAP is GTPase activating proteins. Okay, so how do we turn GTP into GDP? Here, the GAP will use hydrolysis, right? It will get rid of one phosphate group to turn GTP into GDP. Okay, so here, the name is really weird. The GTP activating protein. It seems like, okay, GAP is something that I use to turn it on because it has activating its name. No, it really is the activation of the formation of the inactive form of GTPase. Okay, I hope that makes sense. And then together, GAF and GAP, together they have another name that is called RGS. Okay, so these two these two. Sometimes if you Google the diagram for GTPase, you will see the acronym RGS, which stands for regulators of G protein signaling. Okay, this is not too important for MCAT, but just in case you see it on Google. All right, I hope that's helpful.